Introduction to General Microbiology. Welcome to General Microbiology, the science that studies microorganisms in all their complexity, their structure, functions, growth, control, and their applications in health, industry, and the environment. In this module, we will focus on the basic principles of microbial control, culture preparation, and microscopy fundamentals. We'll begin with sterilization and disinfection methods, move on to culture media preparation, discuss pure culture techniques, and then cover preservation methods. By the end of this first module, you'll have a strong foundation for understanding how microbiologists work in laboratories to safely handle and study microbes. Section 1. Principles and methods of sterilization. Sterilization is the process of completely eliminating all forms of life, including bacterial spores, from an object or environment. It is an absolute term, something is either sterile or it is not. The choice of sterilization method depends on the nature of the material, the type of microorganisms present, the intended use after sterilization. There are two main approaches: physical methods and chemical methods. Control of microorganisms by physical methods. 1. Heat. Heat is the most widely used method because it's reliable, inexpensive, and efficient. Moist heat works by coagulating and denaturing proteins. Common methods include boiling, kills vegetative bacteria and viruses but not all spores. Autoclaving, steam under pressure, typically at 121 degrees Celsius for 15 to 20 minutes, is highly effective for sterilizing instruments, glassware, and culture media. Dry heat kills by oxidation and includes hot air ovens, used for glassware and metal instruments. Incineration, burning to ashes, used for contaminated waste. 2. Filtration. Filtration removes microorganisms by passing liquids or gases through a filter with pores small enough to trap microbes. This is useful for heat-sensitive solutions such as antibiotics, vitamins, and certain culture media components. Common filter types include membrane filters and depth filters. 3. Radiation. Radiation damages microbial DNA, preventing replication. Ultraviolet, UV, light, effective for surface sterilization in laminar airflow cabinets and operating rooms. Ionizing radiation, such as gamma rays, used for sterilizing disposable medical equipment and certain food products. Control of microorganisms by chemical methods. Chemical agents are often used for disinfection or antisepsis rather than complete sterilization. Let's review the main classes you outlined. Phenolics. Derived from phenol, they disrupt cell membranes and denature proteins. They are effective against many bacteria and some fungi. Alcohols. Commonly ethanol and isopropanol, they work by denaturing proteins and dissolving lipids. They are effective for skin antisepsis and cleaning small instruments. Halogens. Such as chlorine and iodine, they oxidize cellular components. Chlorine is used for water disinfection, iodine for skin antiseptics. Heavy metals. Compounds of silver, mercury, and copper can inhibit enzymes and other proteins. Silver nitrate was historically used for newborn eye prophylaxis. Quaternary ammonium compounds. These are cationic detergents that disrupt cell membranes. They are used for disinfecting surfaces and instruments. Aldehydes. Glutaraldehyde and formaldehyde are highly effective sterilants that cross-link proteins and nucleic acids. Sterilizing gases. Ethylene oxide gas penetrates well and sterilizes heat-sensitive equipment. Evaluation of antimicrobial agent effectiveness. The effectiveness of a disinfectant or antiseptic is determined using laboratory tests such as phenol coefficient test, use dilution test, disk diffusion method. These methods measure the ability of the agent to inhibit microbial growth under controlled conditions. Principle and functioning of laminar airflow (LAF). A laminar airflow cabinet is designed to provide a sterile work environment by passing air through HEPA filters and directing it over the work surface in a uniform flow. This prevents contamination of the sample and protects the worker in some configurations. Section 2. Culture media preparation. Microorganisms require nutrients to grow, and culture media provide these in a controlled environment. General principles in preparing culture media. Understand the nutritional needs of the organism. Ensure all ingredients are accurately measured. Sterilize the media appropriately to prevent contamination. Adjust pH to optimal levels for the microorganism. Pour and store media under aseptic conditions. Types of culture media. Simple media. Contain basic nutrients like peptone, meat extract, and sodium chloride, suitable for non-fastidious organisms. Complex media. Contain ingredients of unknown exact composition, such as yeast extract or blood. Synthetic media. Composed of precisely known chemical components. Enriched media. Supplemented with special nutrients, e.g., blood or serum, to grow fastidious microbes. Selective media. Contain agents that inhibit some microbes while allowing others to grow. Indicator media. Change color in response to microbial metabolism. Differential media. Help distinguish between species based on biochemical reactions. Anaerobic media. Provide oxygen-free environments, often using reducing agents. Section 3. Pure culture techniques. A pure culture contains only one type of microorganism. Strict plate method. Used to isolate single colonies by spreading cells over the surface of agar. Pour plate method. Diluted samples are mixed with molten agar and poured into plates. Spread plate method. Diluted samples are spread over the surface of agar plates using a stirrer spreader. Maintenance of pure cultures. Regular subculturing onto fresh media to maintain viability. Preservation methods for microbes. Refrigeration, short-term preservation. Deep freezing, long-term storage at very low temperatures. Lyophilization, freeze-drying for indefinite preservation. This is the end of Module 1. It covered microbial control, culture preparation, pure culture isolation, and preservation, everything a microbiologist must master before actually identifying and analyzing microorganisms.